Hey, Mr. P here. In this video, we're going to talk about prokaryotic cell structures, but those structures that are inside of the cell membrane. So in the last video, if you remember, we talked about all of the prokaryotic cell structures that are outside of the membrane, like the glycocalyx and slime layer and stuff like that. Now we're going to dive in through the membrane and talk about what the structures are, how they're structured, why they're structured the way they are, and what functioning they give to the prokaryotic cell. In a future video, we'll kind of follow this up with eukaryotic cell structures, both inside and outside of the cell. But for now, we're going to focus on just prokaryotic cell structures that are inside the cell membrane. Again, these are bacteria only, no eukaryotes. So, before we talk about what kind of structuring is inside of the cell, we need to talk about what the cell membrane is. And the cell membrane is a structure that's found in both gram-negative and gram-positive bacteria. Okay, remember that we had a gram-positive bacterial cell wall that was on the outside of one membrane. And then we had a gram-negative bacteria which had actually two membranes which were sandwiched, kind of sandwiching the cell wall. Okay, so regardless of the prokaryote, they have a membrane, and we need to talk about what the membrane structure is. You've seen this structure, the plasma membrane or lipid bilayer, in probably previous bio classes. But the membrane is a boundary that consists of phospholipids that make a bilayer. Okay, remember, heads are oriented out in both directions, tails are oriented in. That's because the inside is nonpolar and hydrophobic. The heads are polar and hydrophilic. So that allows a structural adaptation which will allow the lipids to form a boundary between two aqueous solutions. Okay, there is water inside and outside of the cell. There is no water inside where the tails are. Okay, that is a zone of hydrophobicity. There is nonpolar fatty acids in that particular zone. We do not want water, and we'll talk about how water gets in a minute. Inside of the membrane, there are a bunch of embedded proteins. Some of the proteins are what we call peripheral proteins, and others are called integral proteins. All of these proteins that you see here are integral proteins because they are completely integrated into the thickness of the membrane, and they span the entirety of the membrane. A peripheral protein would be a protein that is sitting in just like this, where it only spans one side or only reaches one side of the membrane. A lot of membranes like that are going to be kind of your receptors that are going to receive signals and stuff like that. A lot of your channel proteins, a lot of your glycoproteins, things like that, those are going to be all integral protein. Okay. This is just kind of a close-up diagram of what the phospholipids look like. Remember, the phospholipids consist of a polar region that is a hydrophilic head. Those are polar. They're made of phosphate. And then a phospholipid has two nonpolar fatty acid, which are hydrocarbon chains, which are hydrophobic. Okay, this area right in here from head to head, that's going to be a zone of hydrophobicity. It's going to create a zone that repels water, and all the water is going to be up here on the outside and totally inside of the cell, which is going to interact with just the heads, not the tails. Okay. One thing that is really nice or good for the cells, specifically talking about the cell membrane, is that these lipid bilayers are very fluid. Okay, we use the fluid mosaic model as a model to describe the components and the functioning of the cell membrane. And what we mean by fluid mosaic model is that it is a mosaic. It is a larger structure made of a bunch of smaller subunits. But it's fluid because it has the ability to move. It's flexible. It's not rigid. And the proteins that are embedded look like they're kind of rigidly, structurally sound, but they're not. They have the ability to move freely, kind of laterally, anywhere within that membrane by just allowing the phospholipids to kind of move around them. Phospholipids are the same thing, the same way. Phospholipids rotate and move laterally along the membrane. The only thing that phospholipids cannot do is they cannot move vertically, meaning this particular phospholipid cannot flip to the other side of the membrane. Why? Because the polar head would have to move through the nonpolar region in order to make that flip happen, and it can't do that. But this particular phospholipid, if we're looking here, can move laterally and end up 20 seconds later here in the phospholipid bilayer. So these lipids are constantly moving past each other. They're constantly flowing, if you will, and they're allowing these proteins to move freely within them as well. So the phospholipid bilayer is a very secure boundary, but it is fluid and it does allow for movement. What are some of the functionings or the functions of a plasma membrane? It is a physical barrier. Okay, it's a physical barrier between the outside and inside of the cell. It forms the physical barrier between the inside and outside of the cell. It keeps things that are outside out, and it keeps things that are inside in. The plasma membrane has what we call selective permeability. Selective permeability means that it selects certain substances to come in while disallowing certain substances to exit. It's really important to understand that cells don't just want random things flowing in and out of their cell. They want to keep the things that they have in, and they want to keep things, for the most part, out. There is instances when they want to move things in, and there are instances when they want to move things out, but that's why they want to have selective permeability, so they can select the things that they want to come in and keep the things out they don't want to come in. So some membranes have photosynthetic pigments on foldings called chromatophores. The chromatophores are specific to certain bacteria, and they are going to allow for the process of photosynthesis to take place in unicellular prokaryotes. Obviously, photosynthesis is a process that we are familiar with. It is a process that plants use to generally synthesize energy. But prokaryotes can do that as well. Some prokaryotes can do that as well, and they use chromatophores to do that. Membrane integrity can become compromised by the addition of these substances. So alcohols, quaternary ammonia, which are detergents, and polymyxin, specifically the antibiotics, cause leakage of cell contents. And what that basically means is you have a viable, normal, healthy E. coli cell. When you put some of the polymyxin antibiotic into the environment that the bacteria is trying to live in, it actually disrupts the membrane and kind of drives a wedge, so to speak, into the membrane so that it allows for cell leakage, meaning things can now kind of more easily leak out of the cell membrane because we kind of opened up a portal by which contents can move through. And that causes kind of what we call lysis, and the cell is not viable at that point. So it is a good property of some of the antibiotics we have and is very conducive to cell death or fostering cell death. Okay, but it's specifically tied to the cell membrane. So we talked about the plasma membrane being selectively permeable. Okay, some terms that you've probably heard before are passive transport and active transport. Okay, the plasma membrane is actively involved in the transporting of substances because it is the physical barrier between outside and inside. But some of the passive transport mechanisms that the cell uses, whether it's prokaryote or eukaryote, is simple diffusion, facilitated diffusion through specific transporters. Okay, this would be non-specific transporters, this would be specific transporters, and then osmosis. All of these are passive transport mechanisms, and what that means is they can move materials passively with a concentration gradient without energy, and that's the important part. Okay, with the concentration or following a natural concentration gradient is always in the same direction. It's high concentration to low concentration through sometimes a channel protein, sometimes not, okay, in the case of diffusion, but it always goes high to low and it always moves without energy. So if we dive into these specific passive transport mechanisms in more detail, simple diffusion is the most common simple transport mechanism or passive transport mechanism the cell can employ. It doesn't require proteins. The cell is able to move very small molecules that are typically uncharged through the membrane without channel proteins. Okay, you will see that there is in all diffusion situations a high concentration and a low concentration. Now the high concentration can be outside like this diagram represents, or the high concentration can technically be inside, but you have to have a high concentration and you have to have a low concentration. The solutes will naturally flow through the membrane down their concentration gradient from high to low without the use of energy. Okay, and it will continue to move until the net movement is zero. At that point, equilibrium is reached, and what that means is you have equal concentrations on both sides. When you do have equal concentrations on both sides, the net movement is zero, but the diffused solutes don't stop moving ever. Okay, if we have a membrane and you have equal concentrations of solutes on each side, in this case we have 5 solute particles, net movement is zero because the overall diffusion path has been stopped, right? There is no high and low concentration anymore. But as soon as one moves across this way, then all of a sudden now this side has 6 and this side has 4. So then this becomes the high side and it moves back to the low side. So now it has established the 5 and 5 again. Another one might move this way, and now this has 6 and this one has 4. Well, then this one's going to move this way, and you can see that it's just going to kind of jockey back and forth, always maintaining equilibrium once equilibrium is reached. When we talk about our facilitated diffusion through non-specific transporters, these are things that are going to move through a channel protein. Okay, channel protein is required. That's what facilitated means. It is diffusion down a concentration gradient from high to low, just like regular diffusion, but it is facilitated or helped by a non-specific channel protein in this case, in order for these particles to move through. Okay, these particles are polar or hydrophilic charged particles. And if you remember from the previous slide when we talked about what the zones of this membrane are, you know that the polar and hydrophilic regions are on the outside of the membrane, while this interior is nonpolar. And that tells you right there that if we have hydrophilic polar molecules, or if the molecules are charged, they are going to be repelled by this interior of the membrane. So if they try to diffuse through, they're going to be repelled by the interior of that membrane, which again is why we have to employ this nonspecific transport protein, which will allow these charged hydrophilic and polar molecules to move through without contacting the interior of that membrane. Again, it's a passive transport mechanism. It does not require energy, and it is going to flow in one way from high to low down a concentration gradient until, again, equilibrium is reached. Okay. Now we can look at the difference between facilitated diffusion, but this time it is specific facilitated diffusion. This type of facilitated diffusion still moves down a concentration gradient. We would still have a high side. We would still have a low side. We would move solutes down the natural concentration gradient from high to low, again without energy. But in this case, we are looking at transporting larger molecules like glucose. It is a monosaccharide. And because it is a very large molecule, it can't simply squeeze through any kind of non-specific transport channel protein. It has a very specific shaped large protein that is designed to transport large molecules like glucose. Okay, it has to be the right shape. And it kind of opens up and allows these glucose molecules to squeeze through because it's built to allow glucose to move. Okay, that's a very specific molecule to move. Again, it moves high to low down a concentration gradient without energy, and will do that until, again, equilibrium is reached. The last one is osmosis. Okay, osmosis is specific to water only. Water is a polar molecule. Again, we're going to move, because it is a passive transport mechanism, from an area of high water to low water. Water will move through a specific channel protein known as an aquaporin, and will continue until equilibrium is reached. Now, when we talk about water, equilibrium is actually going to be referred to as isotonicity. Okay? There are terms like hypertonic, hypotonic, and isotonic. Hypertonic is when you have a higher solute concentration and a lower water concentration. Hypotonic would be the opposite. Hypotonic would have a low water or a low solute concentration and a high water concentration. And then isotonic is when you have equal solute and equal water concentrations on both sides of a membrane. Osmotic pressure is the pressure needed to stop the movement of water across a membrane. And cells need to have an osmotic pressure to kind of help balance the water movement into and out of a cell. But osmosis is very simple and straightforward. Again, you've covered this in previous bio classes, but I'm going to just kind of highlight it here. Right? Don't forget about osmosis. Osmosis is one of those critical mechanisms that definitely belongs in the passive transport mechanisms because it moves water passively, not actively. Okay? Again, osmosis, if we have a cell that is equal in solute concentration to its environment, the net movement of water is 0, and water will move in and out of the solution and in and out of the cell equally. If we put cells in a hypotonic solution, meaning the solution that the cells are bathed in has a lower solute concentration than the cells, meaning the cells are more concentrated than the water than the solution is, then water is going to move from the environment into the cell, and that's going to cause the cell to rupture, which is called osmosis lysis or osmotic lysis. We've talked about that in previous chapters as well. Osmotic lysis, that actually kills the cell. If you put a cell into a hypertonic solution, meaning we put a cell that is hypotonic in comparison to its environment, into a really high concentrated or really concentrated solution, it's actually going to lose water and it's going to become kind of what we would consider dehydrated, and the water will move out and it will kind of shrivel up. Okay, that's called plasmolysis. That's osmosis. Plasma membrane active transport mechanisms. So active transport mechanisms is still a way for a cell to move solutes across the membrane, but in active transport processes, we are going to require a transport protein or a protein pump. Okay, in this case, this is the sodium-potassium pump, and it requires ATP, which is energy. Okay, solutes have to travel against their natural concentration gradient. So if the natural concentration gradient is high to low, in active transport mechanisms, they require energy and pump proteins in order to pump the solutes against the natural flow, and so they're going to be moving low to high in an active transport mechanism. Okay. Active transport mechanisms kind of continued. You have what's called group translocation. Okay, this again requires a transporter protein. Solutes are going to move against their natural concentration gradient again. They require energy. It's no different than any other active transport mechanism, but in this case, it's designed to move substances in, do something to the substances so that they are trapped inside the cell so that they can't be lost or then can't transfer back into the environment with a passive transport mechanism. Okay? So the way that this group translocation works is that it is a special form of active transport. It occurs exclusively in prokaryotes, so this is not a eukaryotic process. This is specifically a prokaryotic process. But once the substance, like glucose in this case, moves into the cell, this being the outside and this being the inside, once glucose is brought in by the cell, it's actually going to be slightly altered from glucose to glucose 6-phosphate. And once it does that, once it's altered, it remains inside the plasma membrane and is therefore making it so that it is not going to be able to escape. When we bring these molecules in, or when prokaryotes bring these molecules in, they are going to slightly molecularly rearrange them to make the cell membrane impermeable to the kind of product that it made so that it doesn't have to fight to keep the molecules in. It can simply keep them in based on the fact that the cell membrane will not allow them to move through. Okay? It enables the cell to accumulate various substances even though they may be in very low concentrations outside of the cell. Okay? And the reason I bring this in, this is an active transport mechanism. So if we have one glucose outside, and let's say that in this case it's very low concentrated, we have very low amounts of glucose in our environment, and let's say that inside we have very high amounts of glucose concentration. Well, the natural path or the natural diffusion path wants to be glucose to go from high to low. So glucose wants to naturally diffuse out. The cell doesn't want to lose glucose because glucose is valuable, especially if they're in a solution or in an environment that has low external glucose to begin with. The prokaryotes want to keep the glucose in-house. And so the way they do that is through this group translocation. They bring the glucose in, okay, through a transporter protein, which is designed to transport glucose into the cell. And once the cell takes in the glucose, it's going to use this PEP, which is an energy source. It stands for phosphoenolpyruvic acid. And it's going to use a series of intermediates to phosphorylate the glucose, meaning we're going to add a phosphate to it. Once it has a phosphate added to it, it becomes glucose 6-phosphate, and at that point, it just by itself cannot pass through the membrane because these transporter proteins are specific to glucose, not specific to glucose 6-phosphate. Once the glucose has been brought in and turned into glucose 6-phosphate, it no longer can get out. That's important, okay? Glucose is essential to the functioning of cells, and prokaryotes are no different, all right? What are some of the other internal prokaryotic structurings? Obviously, we need to talk about cytoplasm. Cytoplasm is the structure or is the fluid that all of our internal structures are housed in or bathed in. It is the fluid that is primarily water, okay, roughly 80% water. The other 20% is made up of enzymes, proteins, carbohydrates, glucose, lipids, ions, the cytoskeleton components, and all the other organelles. Okay. When we talk about cytoskeleton, I know you've heard that term before. There are different cytoskeleton components that all do different things in a cell. Again, we are not talking eukaryotic cytoskeleton components. We're talking prokaryotic cytoskeleton components. There are all 4 of these different types of cytoskeletons. So MREB, PARM, CRES, and FTSZ. All of these make up different cell cytoskeleton components that all do different things. Like the PARM is all about plasmid segregation. So plasmids are circular rings of accessory DNA typically associated with antibiotic resistance or viral pathogenicity, virality, stuff like that. Well, these PARM actin-like substances actually help to hold the plasmids apart so that they're easily segregated. The FTSZ, that's your tubulin-like structures. They kind of go around the cell on the outside, and they help to allow the cell to constrict in certain points. You have the MREB. Those are actin-like substances. They help to give the cell its shape. And a variety of other reasons. We're not going to really dive into that, but you have different components that kind of are the microfilament, microtubule, and intermediate-like components. Again, the microfilaments, the intermediate filaments, and microtubules, those are all about kind of the eukaryotic cytoskeleton components, but the prokaryotes have cytoskeleton components that resemble those eukaryotic components. Again, the cytoskeleton assumes roles in cell division, cell shape maintenance, DNA movement, protein targeting, and alignment of organelles. All of those things need structure. The cytoskeleton helps to provide the structure to the cell from the inside of the cell. Okay, moving on to the nucleoid containing DNA. Again, prokaryotes do not have a nucleus. They have a nucleoid region, and they contain DNA in their nucleoid region. Again, you will not see a membrane around this nucleoid region. It is not an actual nucleus, but it is the place where the bacterial chromosome is kept. And bacterial chromosome is circular. It contains no plasmids because the plasmids are kind of kept outside of that nucleoid region. It makes up 20% of the total cellular volume, so it is a relatively large structure. It does not contain histone proteins, so we call that prokaryotic DNA naked, whereas eukaryotic DNA, like our DNA inside of our nuclei, possess histone proteins which help to supercoil our DNA and keep our DNA kind of free of tangles and organized the way that it does. And again, not surrounded by a nuclear envelope, so there is no actual nucleus. Plasmids, I said that is a circular ring of accessory DNA. It is small in comparison to the nucleoid containing DNA. It has extra chromosomal genetic elements. It is not connected to the main chromosome. Like I said, it's accessory. Okay? It typically contains 5 to 100 genes, and they're not usually crucial to the survival of the bacterium under normal environmental conditions. However, they can contain genes that give antibiotic resistance, tolerance to toxic metals, and production of toxins, as well as the synthesis of specific enzymes. I want you to kind of think about the nucleoid containing DNA. That's all about the actual kind of normal cellular activity. Okay, the nucleoid DNA gives the instructions for how to be a normal bacteria cell. The plasmid give directions on how to be more tolerant to substances in their environment, produce toxins, be antibiotic resistant. Okay, all of those kinds of things. Focusing on the ribosomes, in a prokaryote, they are 70S ribosomes in comparison to eukaryotes, which are 80S ribosomes. You can see that a 70S ribosome is going to consist of a larger 50S subunit and a smaller 30S subunit. That gives us a total 70S ribosome. The ribosomes are sites of protein synthesis, so they are going to use the mRNA, rRNA, tRNA to produce proteins by linking peptides, forming polypeptides. Once the proteins are made, they are going to be sent to the rest of the cytoplasm, different not organelles. Okay, prokaryotes don't have organelles, but they're going to be sent to different substances or different areas within the cell, and are going to basically allow the prokaryote to function the way that it needs to do. Okay. Some of the antibiotics that we use, specifically streptomycin and erythromycin, are going to be specific in targeting prokaryotic 70S ribosomes. Streptomycin and gentamicin are antibiotics that will specifically attach themselves to the 30S small subunit, and erythromycin is a specific antibiotic that will attach itself to the 50S subunit. What do you think the benefit of having a very targeted approach, antibiotically speaking, that targets ribosomes? If we target ribosomes and shut down the ribosomes, then we can't produce protein. And if the bacteria can't produce protein, it can't function the way that it needs to, and it dies. So one of the really good ways of inhibiting protein synthesis in prokaryotic ribosomes is the employment of antibiotics that attach themselves to the antibiotic kind of specific regions of these particular ribosomes. Now, I know that if you look at this and if you're good at math, you would see that 50S and 30S look like they should make 80S, but that's not the case. Okay, this is not math. It's not basic addition. The S refers to Svedberg units, which indicates the relative rate of sedimentation during ultra-high-speed centrifugation, which basically means that the 50S and 30S is based primarily on the weight and shape of the actual particle and not additive. Okay, it's not the same as saying 70 plus 30 to get 80, or 70 plus 30. Okay, this should be 50 plus 30. It's not the same as saying 50 plus 30 to get 80. They're just making 70S ribosome. Inclusions, one of the last things we're going to talk about are the inclusions. So inclusions are things that are internal to the cell. They are the vesicles and vacuoles, so to speak, of the prokaryote. Different inclusions would be metachromatic granules or volutin. These are your phosphate reserves. You have polysaccharide granules, that's energy reserve, lipid and sulfur granules or lipid inclusions. These are energy reserves as well. Typically, the sulfur granules are going to help to be energy reserves to help make and structure proteins. Lipid inclusions and polysaccharide granules, those are all going to be able to make and reserve glucose, make energy. Carboxysomes, that is your Rubisco enzyme for CO2 fixation during photosynthesis, only in bacteria that can photosynthesize. You have gas vacuoles, which are protein-covered cylinders that maintain buoyancy. It gives them, it's kind of analogous to the swim bladder of a fish. Okay, it gives them buoyancy in the water. because prokaryotes live in aqueous solutions, they have to be able to be buoyant so that they're not having to fight their environment. And then you have magnetosomes, which are iron oxide inclusions, which are designed to destroy H2O2, which is hydrogen peroxide, which is a byproduct of some of the cellular metabolism that these cells have to go through all the time. Okay? Magnetosomes also kind of resemble or act as a magnet, which can help it orient itself in its environment as well. Kind of its internal compass. Last thing, other internal prokaryotic structures are the endospores, and endospores are present in resting cells. They are produced in certain bacteria when environmental conditions are not as favorable. It basically means that certain bacteria have the ability to produce these endospores when it senses the environment losing different resources or becoming less water, like a lower water concentration, lower O2 concentrations, maybe higher CO2 concentrations. These endospores are incredibly resistant to desiccation, heat chemicals, and radiation, meaning they can withstand pretty much anything. They are very hard to kill. They're produced by Bacillus and Clostridium bacteria. Sporulation is the process of actually producing the endospore, and germination is the endospore returning to the vegetative state. So basically, bacteria can sense, certain Bacillus and Clostridium bacteria can sense environmental conditions waning or becoming less favorable. When it senses that, it goes through the process of sporulation, it makes the endospore, and it waits for a long period of time until the environment becomes more favorable, and then it can return through germination to its vegetative state and produce viable bacteria even after long periods of time when the environmental conditions were not favorable. How does this process work? Inside of a bacteria, you have kind of a budding process that takes place where the spore septum begins to isolate newly replicated DNA. So kind of prior to this entire step, the DNA would have had to replicate itself. A new replicate would have been made, and it's kind of put into the polar region of this bacteria. It's called a spore septum, and it begins to isolate the newly replicated DNA, and it then kind of walls off the cytoplasm to create what will eventually be the endospore. Plasma membrane on the inside of this cell wall is going to start surrounding the DNA, cytoplasm, and membrane that was just isolated in step 1. Then you have the spore septum surrounded and totally isolated, forming the forespore. It then forms a cell membrane and becomes an actual endospore, but it's still kind of contained within the bacteria. The spore coat forms. The spore coat is what actually helps to keep it 100% protected, okay, from the desiccation and the heat or the radiation, and it can then be freed from the cell, and it can withstand a lot of environmental conditions that are not favorable. Okay, you can send radiation at it, you can send a lot of heat, you can try to kill it, you can put chemicals on it. A lot of times you will not kill it. When environmental conditions return, then it germinates and it becomes a viable bacteria again. Okay, that's where we're going to end it. I know this video is a little longer than usual, but a lot of information for that internal structuring of a prokaryote. So bring your questions to class. I'll see you later. Bye.